Can you hear me? Hello. Hello, hello. Can you hear me? If you can hear me, you can leave a message. Not sure if they can hear me. Or you can turn on this. So we don't have this. Should be on. No. Hello. Maybe no one's listening. Oh. Okay, okay, good. That's great. So maybe we'll wait till 3 o'clock to start. We'll wait for the in person. There are people coming from the other group to come. Then we'll start the live session. Okay. Hello, everyone. We are the Advanced Biophotonics Imaging Lab, and I'm Eamon. It's my pleasure to introduce you our lab. So, firstly, the outline for today's lab tour is first I'll give an introduction of our research areas and the main technologies that we use, and then some introduction to the projects that we're ongoing. Lastly, we'll have a demo and showing the results of our experiments. So firstly, our lab focuses on 3 points. First is high performance fiber optic endomicroscopics. Second, we also focus in the image guided operated method. Lastly, we also use novel image interpretation and quantification tools. And in order to achieve these 3 aims, we use the technologies including OCT, which is optical coherence tomography, for retinal imaging as well as intraoperative applications. Second, we also use endomicroscopy and computational optics. Lastly, we use machine learning for the image. So since our lab mainly works related to the optical coherence tomography, so I'll give a brief introduction on what OCT is. It is a high-resolution, label-free imaging modality that can achieve both 2D cross-sectional image as well as a 3D volumetric image based on the biological tissue structures, and it measures the backscattered echoes of light. So when we compare OCT with other imaging modalities like ultrasound or confocal microscopy, OCT shown here has a higher resolution. It means we can see finer details with a reasonable penetration depth. Moreover, we not only can see the surface, we can also incorporate OCT probes into smaller probes that we can visualize inside the body as well. So here are, mainly in OCT, we have a light source where it goes to the fiber coupler and divides into 2 paths, the sample and the reference path. And then when light hits any surface on these 2 paths, it comes back. And then when the distance traveled by the light in these 2 paths are relatively equal, we will get interference at the point, and then we detect the interference with the detector, and then we can post-process into depth result information. So this is a time domain OCT, meaning that in the reference arm, we have a mirror, which reflects most of light, and this mirror is moving. We move the mirror in order to match the length between these 2. However, the time domain OCT is limited since we are mechanically moving the mirror. The speed is limited. So now, more recently, people developed a spectral domain OCT, where the reference mirror is fixed over here. So it has the advantage of a quicker speed, and it's a more commonly used OCT. So here, since it's called spectral domain OCT, we receive the information and we do the Fourier transform in the post-processing, and we can achieve the depth information. And The main consideration of an OCT system are the wavelength and the bandwidth of the light source. It also determines the resolution of the depth resolution of our system. So OCT gives us mainly three kinds of data. First is the 1D axis scan, which is taking one point and we scan. And then if we scan a point transversely, we get a cross-sectional image, a 2D image. And then you take many images around the y direction, you get a 3D. So currently, there are many fields of research for OCT. Mainly, it's also almost a gold standard for retinal images, imaging the back of your eye. However, we can also image the luminal organs. We incorporate OCT into, like, for example, a bronchoscope that we can image the esophagus or the trachea. These OCT images shown here, we can see the cross-section image, and it shows us the structural different layers of these structures. But however, OCT, we can also use different algorithms to process the, for example, here, the angiography. So we can see the blood information too. So OCT can show us both the structure as well as the property information. So now I'll do first do a demonstration on this commercial cell lab system. Then we'll come back to our projects. So this is an application where, so we start, so here's the light source. Light source from here, it's split into here the reference arm, here the sample arm. So we put any sample over here, and then we can see a signal here. So firstly, remember the 1D, we're supposed to see a peak. So we'll wait. Yeah, so here, for example, this plastic cap, and it has some structures underneath over it, so we have a certain depth in it. So, for example, here, we don't see anything, but when we try to adjust the length here, you can observe a peak. Yep, you can see a small peak here. You can see this 8 number here, and then we can adjust the length. Yeah. So you can observe a peak here. It means that the light hits a structure. So there is one layer here. So this is a typical 1D data for OCT. And if you want to take a 2D, you can just here. So this is an 8. We can check a smaller structure. Suppose this 8. Here we can observe. You just imagine we're cutting it like here, and then we're seeing it on the side view. So this is the 2 sides of an 8. That's why we can observe the structural information. Lastly, for the 3D, we can also observe a whole. So these are many D scans, and then we can observe the 3D structure. And we can post processes. Here we can see the 8 here, as shown in the box here. So this is a commercial tabletop system, so we can only see, for example, the surface of anything. Then I'll introduce our existing projects. As I mentioned previously, we have the light source of an OCT system is important. So let's move to that side so we can see the visible OCT system. So here is our visible OCT system, which means the light source, the wave is in the visible light range. It has an advantage. Why we want to use the visible light OCT, because since we have a smaller wavelength, we have a better axial resolution. At the same time, the optical properties of the visible light OCT is better for the angiography, so we can visualize the blood structures. So here you can see we have the light source, and then it comes to our reference arm, sample arm. So this is the tabletop system, where we put a sample and then we can image similar to that, but with a better axial resolution. And over here is the retinal system, which we use to image the eye. So we put our sample here, and then here's the reference arm, and here's the sample arm, here's the reference arm. So I can show you some results here. So this is the surface, you can image from the surface, and this is the setup for the retinal imaging. And here are some results for surface. So we can image the onion, and you can see the cell, large cell of an onion, and in the bottom you can see the finger. You can image the surface of the finger, as well as you can image these pores, like the sweat glands. Here is our experiment for the mouse. We can image the ear, And since the ear is very thin for the mouse, we can also visualize the angiography for the mouse ear. Next, using the retinal system, we can image the retina of the mouse, which we can see the retinal layer. And we can also calculate the unfast image, which is at the top. Yeah. And then we can visualize the blood vessels. Yeah, so that's it for our visible light OCT. So here we can see mainly the surface of it. But we can incorporate OCT into the so here we have the endoscopic OCT. So that's another project that we're doing. Here, similarly, we have the reference and sample map. And then why we want to use endoscopic OCT? Because like cancers like gastric cancer or lung cancer, it can be visualized through the change of the structure inside the organs. And we want a higher resolution as well as minimum invasive. So we want a higher resolution probe as well as a very miniaturized probe. The difference here is that in the servo arm we have this probe that we insert into the luminal organs. Similarly, we have 1D, 2D, and 3D data for these. So here we scan a line, so we have the interferogram that we processes into the PSF, and we can get our extra resolution. Second, we have also done experiments on mouse colon, inserting this probe into the mouse colon, and then we can observe these structural information for each the colon information. Lastly, since it's a probe, it scans with circular. That's why the result for endoscopic OCT is usually the polar view, as shown here. Lastly, we can also render 3D volumes for visualization. Yeah, and then, however, you can see here it's a straight line. And how about we try to control where the probe goes? That's where we come with this OCT robot arm shown here. So here we can use the OCT probe, and it not only allows us to visualize the structural information, we also have an ablation, which means we can burn and destroy the cancer. So this is the robotic arm and its setup. And similarly, we also did the rat colon imaging as the setup here, and then we can observe the different layers of a rat's colon and the 2D, and we also compare it with histology. So, and we also validated that the ablation tip. So this robotic system gives us the flexibility where we can decide where to do the ablation and where to do the imaging. So it has certain rough. Yeah, so that's pretty much it. We have the visible light OCT system, the endoscopic, and the robotic arm. And lastly, we actually after we get the OCT image, we can also do the image processing. Lastly, we also have the automatic image segmentation for OCT, which is machine learning. So here we use networks to train the network to divide the sections of each layer. So as I mentioned previously, the structural information can give us some insight of the cancer or other diseases. So here we can recognize each layer of the OCT image, and then we can also identify the change in median thickness, and then we can identify some disease. Traditionally, these will be used by doctors. Doctors will see many of these itself. So this one is going to be automatic, and it gives a higher precision. Similarly, we have the sheep airway segmentation here, as shown on the B scan, and then we can identify these different layers as well as the green part, which is the blood vessels. So that's pretty much it for our lab demonstration. Thank you, and if you have any questions,